important in chemistry, so spending extra time on this chapter. What distinguishes an organic compound from an inorganic compound? Carbon. Yeah, there's some exceptions. Pretty much carbon. Why? Because there's such a wide variety of carbon-containing compounds. So they separate it from the rest of the periodic table. What features does the carbon atom account for its ability to form so many chemical compounds? There are two features. You know what they are? One, we can form carbon-carbon chains. So that's an important thing. Not just chains, but branch chains, rings, a whole bunch of things. And two, it can form a lot of bonds. 4 bonds is a lot of bonds. Identify the hydrocarbons among the following. Is this a hydrocarbon? No. You should recognize this. What type of compound is that? CH3OH. We saw that earlier. It's called a alcohol. And in fact, this alcohol is methanol or methyl alcohol. How about this one? Is that a hydrocarbon? Yes. This is carbon and hydrogen. In fact, this one's called octane or N-octane. 8 carbons. This one, C6H6, is that a hydrocarbon? Yeah. This one's called benzene. And is this a hydrocarbon? No. Do you know what kind of compound this is? It's not ethyl, but it's kind of similar. There's two amines sandwiching a methylene. This is called methylene, CH2. So we have an amine on one end and an amine on another, and then a CH2 bridging those two. It's an amine, though. It's a diamine. C8H18. Yeah, that's a hydrocarbon. That's octane, actually. All single bonds. Hydrocarbon and carbohydrate are similar terms, but yeah, they're different. Hydrocarbon, a compound containing hydrogen and carbon. When you look at carbohydrate, what is that? Is that a compound containing carbon and hydrate is, stay hydrated is water. And in a way, yes, in a way, no. If we look at a carbohydrate like sugar, you know what the formula for table sugar is? The formula for table sugar is this. It's C12H22O11. And so we got carbon. The hydrate comes from this. Take a look. What is the ratio of hydrogen to oxygen? 2 to 1. And so we have 2 hydrogens for every oxygen. And so it kind of looks like water, doesn't it? In fact, all carbohydrates have a 2 to 1 ratio of hydrogen to oxygen. And so if I pick another carbohydrate, let's pick glucose, C6H12O6. It's carbon, and what's the hydrogen to oxygen ratio? 2 to 1. And so all carbohydrates have a hydrogen to oxygen ratio of 2 to 1. Does that mean there's water present in sucrose and glucose? No, there's no water. And so where is my structure for carbohydrates? Put that here. I must have closed it. And so if we take a look at the sucrose structure, there basically there's no waters there. It just has that ratio, 2 hydrogens, 1 oxygen. Here they're trying to do a wedge dash type thing. This is a planar ring here, benzene type ring, except it has an oxygen rather than a carbon here. This is also a planar ring here. Well, this is a glucose molecule attached to a fructose molecule. So it's bridged right here, bridging oxygen. Distinguish between the structure of an organic molecule as given by its Lewis diagram and the three-dimensional shape of the molecule. Does the Lewis diagram show any three-dimensional? No. The Lewis diagram only shows the connectivity. That's it. It doesn't show the shape. How are molecular structure and molecular shape shown in Lewis diagrams? Molecular structure and shape are kind of the same thing. They aren't shown in Lewis diagrams, right? Just in the 3D. We could draw a 3D Lewis diagram, or in the ball, what they call the wedge and dash diagram. Why can't a zigzag chain of carbon atoms be drawn as a straight line in the Lewis diagram? Because Lewis diagrams need not show the angles. We're going to get a zigzag chain when we have. The other case for a zigzag chain is, let's say if I have a chain of carbon atoms like this. If I look from the top, it depends on the perspective. If I look from the side, I see the zigzag. But if I look straight down, from the top, it doesn't look zigzag. It's going to be straight. But I don't see the dips. If I look straight down at it, it's going to dip down, then dip up, dip down, then dip up. But it's going to look like a straight line, just going straight. So if you walk along this line, you're going to go straight. If you walk along this, you're going to be zigzagging back and forth. Depends on the perspective you look at it. So drawing a straight line is not totally wrong. What is a condensed structural formula? Condensed structural formulas are incredibly useful. So let's take a look at this. What is this here? This molecule? This molecule is ethanol. Ethanol has the condensed structural formula, CH3, which is what do we call CH3? Methyl, and then a CH2, which we call methylene, and then an OH. So we have methyl, methylene, and OH. Or we put methyl and methylene together and we call it this group is called a ethyl group. And so ethyl group can be C2H5OH. And so both of these are condensed structural formulas here. If we do the molecular formula, it looks different. It would be C2H6O. If you look at C2H6O, do you see ethanol? No. It could be, actually. If you look at C2H6O, that might be dimethyl ether. It need not be methanol. But if you see C2H5OH, that's ethanol, and then C3H3CH2OH, that's ethanol. So condensed structural formula is very useful because it tells us how things are bonded. How are alcohols and ethers similar to water in shape and structure? What distinguishes? Well, alcohols and ethers are, are very similar. Here, like for example, alcohol and water is similar, right? In certain ways. And ether is going to be quite similar too, because all an ether is we're going to replace the second hydrogen. So let's knock off this hydrogen, and then let's put in a group here. So if I take off that hydrogen, and I'll stick a methyl here. So throw a methyl on here like this. And then you know what I just made here? I made dimethyl ether. Ether is a COC link like this. And so dimethyl ether. I should kind of draw that up a little bit more, maybe. And so dimethyl ether. And if I look at it, carbon and hydrogen have very, very similar electronegativity, so it's not really going to change the polarity much at all. Those carbon and hydrogen are very close in electronegativity. Dimethyl ether is soluble in water as well. That's a physical property, though. Chemically, they're going to be different. You can drink ethanol, and your body will metabolize it, but if you drink dimethyl ether, it's not good for you. It wouldn't be calories, I think, in a certain sense. How do carboxylic acids differ from other acids, such as hydrochloric, sulfuric, and carbonic acid? Well, carboxylic acids contain the carboxyl group. And so Let's take a look at sulfuric acid. The sulfuric acid contains a carboxyl group. And so let me just show you the two ways we would draw sulfuric acid. One way we do it like the book. If we do it like the book does not like to expand the octet. And so if we do it the book's method, then we're going to get a structure that looks like this. In which we leave it as an octet, but the formal charges are kind of high because each oxygen is going to have -1 formal charge, and the formal charge on the sulfur will be plus 2. And so it's a little high. And then we add those all up, we'll get a net charge of 2-. Versus if we try to minimize the formal charge, we're going to draw. Oh, am I doing sulfuric acid? This is sulfate. Sorry. Sulfuric acid is going to have 2 H's. What was I thinking? Sulfate. So that's going to knock those two off. And so still it's going to have a +2 and -. This whole thing is going to be neutral. Well, I can get rid of those formal charges now. What I do here is I'm going to form some double bonds. If I pick this oxygen to form the double bond, is that a good choice or a bad choice? Why is that a bad choice? Because if I do that, you know this oxygen is going to have how many bonds? It's going to have 3 bonds. 1 double, 1 single. 3 bonds in that sense. And that's going to change its formal charge. It's going to go positive. So I don't want to pick this oxygen bridging. I want to pick these. These are called terminal oxygens. These are called bridging oxygens. So I'm going to pick the terminal oxygens there. And then I'm going to go from 1 bond, because the oxygen likes to form 2. And so we'll go 1 bond, 2 double. We'll get something like this. And so here, what are the formal charges? All the formal charges are 0. And so how do people like to draw sulfuric acid? So let's see the structure. This is pretty much how people like to draw sulfuric acid. They like to draw it with 2 double bonds. You see the double bonds, they're trying to be a little bit more. Okay, so here, 1 leg forward, 1 leg back, 2 arms up, you see? So here, we're going to have the bridging oxygens here. And then the double bonds here. So 3D. So 3D drawing. Sulfuric. This is some kind of safety data sheet from NIH, Library of Medicine. What other structures? There you see 2 double bonds. Now here, does it matter where I put the hydrogens? No, it doesn't, because it's tetrahedral. Tetrahedral, you know, all the positions are the same. Even though I move the hydrogen here, it's the same thing. It's a 109. No matter what, if I move it back here, it's a 109. They're all the same. It's spherically symmetric. It doesn't matter where I put the hydrogens. Even though it looks like the hydrogens are opposite each other, they aren't. They're adjacent, because that's the V that comes out like this. And there's going to be another V going in. So they're all adjacent. And so I can put the hydrogen here, same thing. It's tetrahedral shape. Okay, then one thing though, I was looking at the drawing. Was that your, Isabella? The drawing?
Yeah, I drew sulfate yesterday.
Okay, you drew sulfate yesterday, so that's probably why I was thinking sulfate. But there was one thing I was going to say. You had 2 resonance forms for sulfate, right?
Yeah.
I think you had something like this.
Yeah.
And one more. I think it was like this. Was that it?
Probably.
Okay. So there's just one comment about this I need to make. One, in resonance, actually two comments. One, in resonance form, we never move the atoms. And so once I fix the hydrogens, I leave them. I don't move them. And so I'm not going to be moving the hydrogens around to create new resonance forms. You don't touch the atoms. The only thing you touch are the electrons. And so these are two resonance forms, but how many resonance forms in total do I have? Just two, or are there more? There are more. How many total resonance forms for sulfate are there? Well, are there 6? Okay, so this is how I count it. It depends on the double bond. So top, bottom, that's 1. Then left, right, that's 2. And how many other unique combinations do I have? Well, I'll just stick with this one. We'll go top, left, that's 3. And then top, right, that's 4. And then we have right, left. And then we have top, bottom. And then we could go left, bottom, and then right, bottom. And so that's 1, 2, 3, 4, 5, 6. 6 is right, 6 resonance forms. None of these are, what we're going to observe is we're going to average all 6 to come up with the hybrid, resonance hybrid. And the resonance hybrid should be more accurate than any of the individual ones come up with. Okay, what I want to do is, I need to continue this, but I want to show a video here. This is a video I tried to show that our VCR is broken, but fortunately it's online. It's old. Old video that it's titled Molecular Structure in Health from Berkeley.
Of the Chemical Education Material Study.
The number and variety of living systems is remarkable. More than 1 billion species currently exist. We now know that a principal factor in this large number is the billions of ways that 4 rather simple chemicals can vary their sequence in forming DNA genes. We are also discovering that many illnesses can be identified at the molecular level. This provides information for the design and synthesis of molecules, which can be used to treat the disease. If we examine living cells under the microscope, like these plant cells, or these bacteria, or this animal tissue, we can see the movement, growth, and cell reproduction, which are the result of successions of chemical reactions. In attempting to control these reactions, physicians have used various substances, some from nature, and some synthesized in the laboratory. Many of the compounds found to be useful were discovered by trial and error. One such compound was sulfanilamide. Let's demonstrate the effect of sulfanilamide on bacterial growth. A nutrient medium in a petri dish is used to sustain growth of bacterial organisms. These bacteria, harmful to humans, are carefully transferred from a stock preparation. They are gathered in a wire loop and streaked onto the nutrient medium in both compartments of this dish. A small piece of sterilized filter paper is wet with a sulfanilamide solution and placed on the bacteria in one compartment of the Petri dish. The dish is placed in an incubator for 24 hours. During that period, seen here in time-lapse photography, the bacteria multiply and become visible as large colonies everywhere except in the area around the paper disc. The clear zone shows that bacterial growth has been stopped. This inhibition is due to the sulfanilamide from the disc. How does sulfanilamide stop the growth of these harmful bacteria? First, let's examine the function of one of the key molecules that exists in the bacterial cells.
Okay, actually, let me stop here just to do the color coding. Black, carbon, red, white, blue, nitrogen.
It is called para-aminobenzoic acid. Using a geometric symbol retaining the general contours of the molecule, we'll follow its action within the cell. In the cell with the acid is an essential bacterial enzyme. In order for the enzyme to perform its active role, it must first combine with the para-aminobenzoic acid. Only then can the enzyme catalyze the further reactions necessary to cell life. When we examine the structure of the acid molecule and compare it to a molecule of the drug sulfanilamide, we find that they are much alike. This large portion of each is identical. The electron charge distribution in each molecule is also similar, and their general configurations are the same. Using geometrical representations again, let's include sulfanilamide in the bacterial cell along with the normally present acid molecule. The sulfanilamide too can react with the enzyme. But the sulfanilamide not only fails to activate the enzyme as a catalyst, it also prevents the acid from doing so. Thus, metabolism stops, and the bacterial cell dies. The effect of sulfanilamide on cell growth was found by trial and error. But the discovery that sulfanilamide is structurally similar to an essential molecule accounted for the action of the drug. This principle of structural similarity helps us to predict which drugs might be effective against specific diseases. Consider, for example, the use of drugs in the treatment of cancer. By means of tissue culture technique, human cells are kept alive for microscopic observation. Here are normal human skin cells under the microscope. In time-lapse photography, cellular activity appears accelerated. We know of very few differences between the chemistry of normal cells and these cancer cells. One remarkable difference, however, is that the cancer cells grow faster. And another difference is that during the faster growth, the cancer cells use abnormally large amounts of a substance known as uracil. Can a structural modification of the uracil molecule be used to disrupt the fast growth of the cancer tissue? Let's follow some of the steps a biochemist takes in answering such a question. He is familiar with the visible parts of a single cell, like this one undergoing division. We see a set of dark ribbons in the middle of the cell. These dark objects are chromosomes, and they are intimately connected with the process of cell growth. The biochemist has learned to collect chromosomal material from cells and has found that it is composed of deoxyribonucleic acid, or DNA. Because DNA is a large, complex molecule, it is helpful to break it into its component parts and identify them. These component parts can be separated from each other by the technique of paper chromatography. After treatment with an acid, a small drop of the chromosomal material is placed on this large piece of filter paper. The paper is rolled into a convenient shape for the next procedure. We have selected a solution in which the chromosomal material is readily soluble. This is placed at the bottom of a glass cylinder. The solvent is drawn up the filter paper by capillary action. After 12 hours, the solvent front has risen this far. The filter paper is removed and allowed to dry. It is then rolled in the opposite direction and put back in a different solvent. This will ensure a further and better separation of the molecular components. These components are not visible under ordinary light, but if we place an ultraviolet light under the dried paper, we can see that the solvent has distributed the components of the chromosomal material. This distribution has occurred because the solvent front competes with adhesion to the paper for the chromosomal materials, so the components move according to their molecular size and solubility. Thus, different compounds derived from the chromosomes are separated. We'll cut out one of these marked areas and dissolve the material from the paper with another solvent. Now we can analyze the dissolved material with an infrared spectrometer. A characteristic spectrum is produced by each component separated in the chromatography procedure. The absorption spectrum produced by one of these extracts is compared with the known absorption spectrum of the chemical thymine. Apparently, chromosomes contain thymine. Similar analysis of the other components shows that each has its own characteristic spectrum. Identification shows that besides thymine, chromosomes contain adenine, guanine, and cytosine. How do our uracil molecules, which are present in such large amounts in cancer cells, relate to the constituents we have extracted from the chromosomes? We know that these constituents, along with other building blocks, are necessary to make DNA molecules, which in turn have a part in forming the chromosomes. Biochemists have shown that uracil is the substance from which thymine is formed. Without uracil, thymine will not form, preventing the synthesis of DNA, which in turn prevents the formation of the chromosome. When we examine the molecular structures of uracil and thymine, we find that there is but one position in which these structures differ. To convert uracil into thymine, a hydrogen atom here must be replaced by a methyl group such as this. Let's symbolize uracil with its hydrogen atom like this to follow its action within a cell. First, a chemical group called a sugar phosphate residue becomes attached to the uracil molecule. The modified uracil compound then forms a complex with an enzyme. Somehow the enzyme converts the hydrogen into a methyl group to form the thymine compound. This molecule is then freed for eventual synthesis into the DNA chain. How can we disrupt this mechanism and stop cell growth? Clearly, one point of attack is the original uracil molecule. This molecule can be altered in many ways. One of the most promising has been to replace this hydrogen with a fluorine to give 5-fluorouracil. 5-fluorouracil has a number of advantages. First, the hydrogen and fluorine are almost the same size, so that both molecules should fit into the enzyme. Second, the chemistry of a carbon-fluorine bond differs from that of a carbon-hydrogen bond, so that the enzyme might find it difficult to replace the fluorine with a methyl group. And finally, the changed polarity may allow the fluorine compound to adhere more firmly to the enzyme than would a uracil compound. Thus, we predict that the 5-fluorouracil, just like the uracil, will attach itself to a sugar phosphate residue and then fit into the same place on the enzyme the uracil would have used. But once in place, the strong bond to fluorine will prevent the enzyme from replacing the fluorine with a methyl group. And finally, we predict that the strong polarity of the 5-fluorouracil will cause such strong adherence to the enzyme that the normal uracil compound will be prevented from reacting, and that, with the enzyme blocked, the essential chromosomal material cannot be made, and the cell will die. Let's test this prediction in the laboratory. Here are two cultures of cancer cells. Those on the left have been treated with 5-fluorouracil. Compare the action of these cells to that of the untreated cells on the right. The untreated cells continue to show rapid growth. The 5-fluorouracil treatment is seen to stop cell division, produce cellular enlargement, and eventually contraction and death. But are we sure that the cancer growth is stopped in the manner we predicted? Instead of the 5-fluorouracil blocking the enzyme as we have suggested, and using this means to kill the cell, is it possible that the 5-fluorouracil is incorporated into the DNA, inhibiting its function? To determine where the 5-fluorouracil is acting, in the enzyme or the DNA, let's conduct an experiment on some human cancer cells grown in the laboratory. A sterile pipette is used to transfer the cells into a large flask which contains a nutrient medium. This permits continued growth so that we will have enough cells for our experiment. The cells are kept in suspension by a magnetic stir and incubated for several days. Here is specially prepared 5-fluorouracil containing radioactive carbon-14. By tagging the 5-fluorouracil with these radioactive atoms, we can determine where the 5-fluorouracil goes in a human cell. We'll add a small amount of the radioactive 5-fluorouracil to the living culture. In about half an hour, these radioactive molecules will be incorporated into the cancer cells. The cancer cell suspension is poured into large cups to be spun in a centrifuge. The spinning collects the suspended cancer cells into a pellet on the bottom of the cup. The liquid is discarded. By this means, the cells from large volumes of the culture medium are finally concentrated into one cup. The cells are then resuspended in cold sodium chloride solution to dissolve the DNA. The low temperature retards enzyme action.
Okay, I mistimed this, so there's still a bit left on this video, so what we'll do is we'll just continue this tomorrow.
Double